All right, good morning. So if I can have more people please sit down. We have an important lecture. I'm Aliyah Uch. I'm a pediatric gastroenterologist from University of Iowa. It is my distinct pleasure and honor to present this year Frank Brooks Lecture Awardee. I think I should just... So just a few words about Frank Brooks. He was the co-founder of the APA along with Dr. Goa and Paul Webster in 1969. So many things I can say about Dr. Brooks, but I'm just going to try to keep it very brief because I really want to highlight the speaker today, Dr. Lowe. He was, like Dr. Lowe, he was a leader, he was a scientist, he was a great clinician, but the most importantly, he was a mentor. He really inspired many people in his lab and in his department. And in his honor, APA began the Frank Brooks Memorial Lecture beginning in 2005, and they're all listed here. So as part of this presentation, I asked Mark to just give me some content so that I could speak about him, and he gave me a few slides, and of course he's a wonderful researcher, and he didn't say much about it, but he's a wonderful clinician. He was a pancreatologist, pediatric pancreatologist, actually before pediatric pancreatology existed. He worked in Dr. St. Agnes's lab. For those of you who may not know, he was really a giant in the CF pancreas field. And then he worked on the lipase research, and then he really helped build the INSPIRE pediatric pancreas research. He was the only NPF mentor for a while until more people joined. And he held the longest run in the T32 field in pediatric pancreatology. Many other things I think he contributed as a leader, as an administrator, and he was the first, and I can't just really begin to really describe his contributions. He's humble, he's graceful, and he's now the first pediatric gastroenterologist as the awardee for the Frank Brooks Lecture. So please.
So thank you, Aliyah, for that kind introduction. Thank you to the American Pancreas Association for this honor. I'm not sure who was on the committee that chose me, but whoever you are, thank you very much. And let's see, which one to go? So these are my disclosures. That represents about what they're worth. And I chose my title today because to me it represents the past from which we need to move away from based on knowledge and education. We're doing that. But throughout much of my career, I would see families who had a child with one or more episodes of pancreatitis, who had an encounter in an ED or another office provider, who heard some version of my title. You know, when did your child get into the liquor cabinet? And that has gotten better. Although I must admit, 2 weeks ago, I saw a 7-year-old after a second episode of pancreatitis. They went to the local ED, and he was asked how many beers he had drunk. I hope the physician was trying to be funny. Mother didn't think it was funny. So we need to move through that. And to me, we are moving forward. And because I think some of this, the reason people saw this was a bias that children didn't have pancreatitis. And if they did, it was related to trauma, maybe hereditary pancreatitis, but usually they didn't think of it. And for a long time, adults who had pancreatitis were thought to be using alcohol. And so given those two things, it's not surprising that that's what we encountered. And to me, this started to change. Yeah, let's see, which way does this go? With a single author, single institution study back in 2002 by Jim Lopez that looked at the number of patients admitted for pancreatitis, both new and all cases, for between 1993 and 1998, and showed that that incidence had been increasing. So yes, children do get pancreatitis. When Veronique Morinville was with us when I was in Pittsburgh, we also did a similar study and looked at what happened at Children's Hospital of Pittsburgh between 1991 and 2003. And the number of new cases are in the blue and red, and the all cases are in blue. And one can see that in the early 90s, there were no new cases diagnosed. And that did increase. And as Mysom pointed out, that has sort of leveled off over time and increased. And we asked why. And one of the simple things that we did was just look at the number of lipases and amylases that were ordered and compared those to the number of patients with acute pancreatitis each year. And there was a correlation coefficient of 0.94, which is almost unheard of in free-ranging humans. So I think a large part of that is physicians were becoming more aware. They were thinking about pancreatitis in children who came in with abdominal pain. And I suspect back in the early 90s, they were told they had a virus, go home. You'll be better in a while. And that, I should say, has been replicated in multiple countries and multiple institutions with different types of data. We also now know, through the work of Inspire and others, that kids also get chronic pancreatitis. And this is just a probability curve of progression from their first attack of acute pancreatitis to developing chronic pancreatitis. And in children that had their first attack at 6 years or older, or less than 6 years of age at their first attack. And one can see that if you have an attack at less than 6 years, you'll, on average, develop chronic pancreatitis within 5 years. If you have it later, it'll take about 3 years. So we now know that kids have pancreatitis, they get chronic pancreatitis. But why do they get chronic pancreatitis? And the Inspire group has looked at this, and it turns out that most children who have chronic pancreatitis have a genetic risk factor associated with their disease. Obstructive causes are common. There was a time when gallstone pancreatitis was not thought to occur in children. It does, and at the same frequency or relative frequency that it does in adults. And when we recently looked at the Inspire group of 944 subjects, that also included a clinical group from Cincinnati, my son's group, and looked at genes that are associated with risk for chronic pancreatitis, we found that at least 73% of the subjects had at least one genetic risk variant. Now, some of these are difficult because a lot of these they really are risk variants, and some of them are very common. Even though you can show a statistical association with the risk for chronic pancreatitis, there are a lot of people who have the same variant that don't have chronic pancreatitis. So it's always difficult to know for sure whether that is causing that chronic pancreatitis or not. Some we have better evidence, both from clinical studies or from basic chemical studies. So it's really important for us to have a broader understanding of these genetic variants and what they do and what the risk is. And we have spent a lot of time working on preclinical models of chronic pancreatitis associated with genetic variants. And I'm going to focus on 3 of them today, as 2 of them are from our lab, and the other is from Mikla Sahin-Toth's lab. So the first you've heard about already this morning if you were at the session. This is the MODY-causing mutation, or MODY-8 in carboxyl ester lipase, that we, along with the Anders Mulvin group, looked at. We also look at a preclinical mouse model of misfolded lipase variant that develops chronic pancreatitis. This variant was reported in 2 brothers who had poor weight gain, steatorrhea, abnormal fecal elastase, and abnormal pancrealoral test. If the young folks don't know what that is, they can ask Chris Forsmark after the talk. And those suggested that they might have chronic pancreatitis, but there was never any imaging, and they don't have, and they were lost to follow-up. But we made a mouse model of this particular variant and to see whether or not it causes chronic pancreatitis. In fact, it does. And then the human CPA1 mutation that was described by Mikla Sahin-Toth group. And all of these, importantly, develop spontaneous chronic pancreatitis. You don't have to give them extra cerulean or anything else to sort of trigger the cause, so they develop it. And if you look at the histology of these, the CPA and the PNLP histology looks very similar and alike. The MODI-8 looks different. There's a lot more fatty change. In this case, it looks like there's a lot more ACE intercell dropout. But these resemble the chronic pancreatitis in humans. And we began to ask the question of whether or not PNLP and CPA1 variants have the same pathophysiology. And this was based on, there was in vivo and in vitro data to suggest that these were diseases of protein misfolding. So when we looked at bulk RNA-seq comparison of, from the pancreas of the CPA1 and the lipase mice, we saw that on a principal component plot shown on the left side, that the CPA and PNLP mutant mice clustered together, whereas the wild type or reference controls for each group also clustered together, and there was a clear separation. Now, when we did pathway analysis, shown in the upper right bar chart, upper left bar chart, sorry, we saw that there were some pathways that seemed to be upregulated in one over the other at 1 month of age. But by 3 months of age, there were no pathways that had a false discovery rate of less than 0.25, which is a not very stringent criteria. And that also goes for a hallmark and, surprisingly, gene ontology pathways as well. And this, complete characterization of these have been published, but so I'm not going to go into any more detail with this at this moment. And these are both thought to be protein misfolding diseases, and I show this just to remind us that ACE cells make a lot of protein. There is an inherent misfolding rate when you do this, when you make proteins, and that is dealt with by foldases and chaperones and other things in the ER. And when those proteins accumulate, they trigger what's called an unfolded protein response, which causes downstream effects to upregulate mechanisms to increase folding and also increase degradation of misfolded proteins that can't be refolded. Now, when this system gets overwhelmed, you have a maladapted UPR, and that leads to apoptosis and cell death. And that's thought to be the mechanism. And so when we look at some of the downstream targets of the unfolded protein response shown in these heat maps done at 1 month and 3 months in the lipase mice compared to wild type on your right, and the CPA mice compared to wild type on the top. You can see that there's already evidence of upregulation of these downstream targets at 1 month of age, and by 3 months of age is robust regulation of these downstream targets, indicating that that these may well be diseases caused by unfolded proteins. And in this manuscript, we showed that the pathophysiology of the lipase and the CPA1 mutants were pretty much equivalent. They're very similar, if not identical. But what about MODI-8? One of the things that I didn't, a little bit misleading about the histology I showed you before, is that it doesn't show the complete picture. So because the CPA1 and the lipase mutants we think are very equivalent, and we're going to focus on the lipase mutants, comparing them to MODI-8 from now on. So if you look at a section of the whole mouse pancreas and pancreatic lipase, you can see evidence of cell dropout. It seems to be pretty uniform. The MODI-8 at 3 months looks fairly normal. And this is a view of what the T20, the lipase mutant, looks like at 3 months, and very similar to what I showed you before. But if you look at the MODI-8, you can see areas that look abnormal. So there's a section in the middle of this box that there's a little more space between the acer cells. They look a little different than the ones in the upper right-hand corner. And there's some ongoing damage there. And then there's an area up here where there's considerable damage. So whereas the injury in the lipase model seems to be fairly diffuse, at least at first, the injury in the MODI-8 model is patchy, which suggests that perhaps they do have different mechanisms to get to disease. So we did bulk RNA-seq analysis of MODI-8 and compared them first to wild type. And on the right-hand side there's a volcano plot. I know you've seen a lot of volcano plots about here. And they've all been, a lot of them have been different. So I'm going to explain a little bit about these plots. So first of all, we selected the upregulated genes based on having a false discovery rate of less than 0.05, which means that they're going to be likely to be significant. Now there's a line on the x-axis that's at +2 and -2, and then we made different colors of the dots. Now that's a log fold change, and so that means that those are more likely to be significant. But that placement at 2 is arbitrary, and you can place it at different points, and you've seen volcano curves here that have put them at 1, seen one that didn't have one at all. And you can use the black dots to do a pathway analysis. And on this plot, the plus side is for the MODY8, and the minus side is genes that are upregulated in the wild type. So if you do a pathway analysis at 1 month, what we can see is that epithelial mesenchymal transition is greatly upregulated, and that's signaling tissue remodeling and fibrosis. There are G2M checkpoint, E2F targets, and mitotic spindle pathways that point to cell cycle activation. There's apoptosis and angiogenesis that suggest early death and vascular changes. You have upregulated pathways that are in the innate immune pathways. Glycolysis is up, which can be a hint that there's metabolic stress. What's missing in these is the UPR. So we don't think that the overt ER stress is evident in this bulk data, and it may be because it's confined to certain acer cells and it's diluted in the RNA-seq. In fact, that it's related to the patchiness. So overall, at this point, the pancreas is entering a remodeling phase with fibrosis, proliferation, and metabolic stress. Well, immune activity is present, but it remains secondary. If we now look at 2 months of age, once again, we see that there are a number of hallmark pathways that are upregulated. And I'm going to say something else that I forgot to say at the first slide. One, we've also done other pathway analysis, GRACTM, KEG, and genetology analysis, and they are consistent with what we see with the hallmark. The other thing is I should point out that each of these pathways most of them have branch points with different endpoints. And one can get an idea, a sense of what of those pathways and branch points is driving the upregulation of these pathways by looking at what are called leading edge genes, or the genes that are statistically driving this comparison. And we have done that. And so some of the conclusions I'll say are really based on that, but to save time, I'm not going to show you that data. So now at 2 months, we start to see more inflammatory escalation on that continued remodeling background. The newly upregulated pathways of INF, alpha, TNF, kappa beta, IL-6, STAT3, complement and allograft rejection signal a strong immune cell influx and strong cytokine activity. And those signals can drive matrix remodeling and proliferation through STAT3, MYC, and P13K, mTOR axes, which we see are upregulated. So this marks a shift from the adaptive remodeling that we saw at 1 month to more of an inflammatory injury. By 3 months, we start to see clear escalation of stress and inflammation and some hallmarks of chronic injury. There is now strong upregulation of the UPR and hypoxia pathways that signal cellular stress. There's increased MYC targets, TNF, NF-kappa B activity point to rising proliferation and inflammation. We also see continued apoptosis and stress signaling through mTORC and P53 and tissue remodeling via the epithelial mesenchymal transition. The angiogenesis and cholesterol homeostasis are up, indicating vascular and lipid metabolic changes. Overall, the profile now reflects chronic stress, immune activation, and metabolic dysregulation in a stress-adapted state driving progression. So if we put this together and compare it to what we know from T21 and things that are already published. So if we look at 1 month, the MODY8 shows early tissue remodeling and proliferation, whereas the lipase model showed early acenter injury and innate immune response. By 2 months, there's a superimposed immune cytokine surge. But also in the T221, the inflammation intensifies, but there's an expansion of acer ductal cell metaplasia and the epithelial mesenchyme transition and fibrosis. By 3 months, we start to see chronic injury, stress, and vascular adaptation, whereas the lipase model at 3 months is dominated by a fibroinflammatory consolidation and epithelial reprogramming. So suggesting that the damage and injury here is going through different pathways. So we decided to compare, because the progression of MODI-8 seems to be a little slower than T221, we also looked at 6 months at MODI-8. And in this, we also see some of the same things that we see at the earlier months. So by 6 months, the disease has advanced to full immune inactivation fibrosis, and now we see a lot of adipogenesis or fatty replacement. Multiple inflammatory pathways are up, both in the innate and adaptive immune responses. Cellular stress and cell death persist while fibrosis expands alongside the metabolic reprogramming, which includes the adipogenesis I mentioned. So overall, this pathway now reflects chronic pancreatitis with immune activation, stress, and death, a metabolic imbalance, and fibrosis. These insights and further work on leading-edge genes can guide mechanistic studies and therapeutic strategies targeting inflammation and fibrosis, which is probably active in the ductal, stromal, and immune cells. So the working disease model for the MODI-8 pancreas at 6 months is that we have a C8 intercell proteotoxic ER stress, activation of inflammation, acinar ductal metaplasia with KRAS signaling, PSC activation and fibrosis, fatty replacement, and proliferative repair are all indicated by this. So now we went on to compare the MODI-8 to the T21, the lipase mice at various ages. So at 1 month, the two models mainly have unique upregulated pathways. And what I'm showing this now, I've sort of changed this up to keep you on your toes. On the your right is now a bubble plot that shows the pathways on the y-axis and the minus log 10 false discovery rate on the x-axis. And the size of the bars represents the fraction of genes in the gene set for that pathway that are upregulated. So it's the upregulated genes divided by the total number of genes in that pathway. So the bigger the bubble, the more genes in the pathway were significantly upregulated. MODI-8 are in blue, and the lipase are in orange. And one can see that there's very little overlap here. MODI-8 is dominated by OXPHOS and fatty acid metabolism with reactive species pathways, mitochondrial adaptation and oxidative stress. MYC and mTORC suggest energy boost under stress. T21 is the upregulation of cycling G2M, mitotic spindle, EMTA apical junction, and active proliferation and epithelial remodeling, suggesting early fibrosis. There's metabolic rewiring. MODI-8 shows mitochondrial respiration and stress with counterregulation. T21 shows glycolytic shift typical of activated fibroblasts and cycling epithelia. The inflammation is present in both. And interestingly, we see in MODI-8 that the beta cell gene set is upregulated. You heard this morning that they don't develop diabetes, but this suggests that there may be some effect on beta cells in this model. T21 shows stress-driven inflammation via P53 apoptosis, UV response down in cytokine signaling. P53 is the only shared pathway, but its role varies in the two models. So you look at the leading edge genes, MODI-8 links to mitochondrial and reactive oxidative stress, whereas T221, P53 is modulating cell cycle checkpoints and apoptosis under proliferative stress. So in summary, the MODI-8 shows a metabolic mitochondrial stress and innate immunity, where at this age, T221 already shows proliferation, epithelial remodeling, early fibrosis, and glycolytic reprogramming. If we now jump to 3 months, what we see is that the significantly upregulated pathways are mostly unique to each model at 3 months. There are several pathways that are in common. Once again, MODY8 is still dominated by mitochondrial metabolic programs, whereas the T221 is still being driven by fibroinflammatory and proliferative programs. So there's there's distinct differences between the MODY8 and the lipase T221. So if we then went and compared, because of the differences in progression, we compared the 6-month MODY8 to the 3-month lipase model, and one can see in the bubble chart that now there are a lot more overlapping pathways, although the upregulation in the pathways are different, particularly in the bottom with the epithelial mesenchymal transition, that many more genes involved in the lipase mutant than the MODY. But there still are some unique signatures that indicate distinct biology. So in the lipase model, the pathways tied to tissue remodeling fibrosis still dominate. So it has a strong fibrotic phenotype, and MODY8, the unique pathways still reflect proliferation and lipid signaling consistent with fatty change. Some of the shared pathways, as I said, differ in enrichment strength, indicating different gene contributions, and overall T21 emphasizes remodeling of fibrosis, and MODY8 shows sustained immune activation, a background of proliferation and fatty change. I know I'm being repetitive, but that's seen over and over again. And if we now look at ACER enzyme production at the 6-month MODY8 versus the 3 months lipase mutant, we see that they both show reduced overall expression compared to wild type, but the magnitude is greater in the T2 in the lipase mutant with a mean T of minus 14.5 compared to minus 2.8 for MODY8. And all of the enzymes except trypsin 5 are significantly decreased from wild type levels in the lipase mutant, whereas only colipase and elastase, various elastases and CTRCs are significantly different between MODY8 and mild type. And this is findings consistent with more chronic ACER injury and dysfunction at this age in the lipase model. So if we compare the two models, what we can see is that the lipase mutant progresses faster. It's pro-fibrotic. It has innate immunity, ductal metaplasia. The lipase has greater decrease of enzyme expression, so faster, more progressive acinar cell injury and cell loss. MODY8 has increased adipogenesis and lipid metabolism. There's fatty change and metabolic alterations. And interferon and antimicrobial injury signatures are upregulated in both models. So this has model-specific implications for the mechanism. So T221 is an early global injury phenotype with acinar collapse, ductal metaplasia, fibrosis, and strong immune response. Pathology in the microenvironment predominates. Acinar cells appear to carry major burden dominated by proteostasis and mitochondrial redox changes. Fatty change is much stronger than fibrosis, and the pathology in the microenvironment is limited. Thus, the lipase and CPA models may be useful for interrogating the connections between fibrosis, immunity, and epithelial cells, whereas MODY8 may be a useful model for interrogating ACER metabolic and ER stress biology. So this could also provide some therapeutic thoughts. MODY8, as I said, is an ACER metabolic stress. The lipase is a fibroinflammatory proliferation. So one may be able to address ER stress modulators and mitochondrial reactive oxidative species buffering, mTORC1 modulators or fatty acid oxidation, peroxisome flux modulation. And there are known medications that can address all of these. And whereas in the lipase, fibrosis inhibitors inhibit innate immune pathways, and targeting hypoxia glycolysis metabolism or the cell cycle may be targets. But in reality, I don't think that any of these are going to be the answer in these models. Both of these our systems where a defective product is made and misfolded protein. And in order to treat these, you either have to decrease the amount of defective product, increase its destruction or degradation, or repair it through, in this case, through proper protein folding. And I think those are the approaches that eventually will be helpful in these diseases. I just want to take a minute for something I didn't say. All of us have people that have touched us in one way or another. And I want to acknowledge some folks who are actually no longer living. So Dick Grand, Peter DeRee, who introduced me to the CF and the Shwachman-Diamond syndrome communities, as well as to the PERT communities. And I also would like to, I guess Dave Whitcomb isn't here this year, but I'd like to thank David because he actually introduced me, drugged me to the board of directors. I'm not sure the NPF knew I was coming. And also gave me the opportunity to introduce a pancreatic program, a pediatric program at Pancreas Fest, which did a lot to stimulate work here. And finally, the people that were stimulated are now largely part of the INSPIRE group, which has contributed so much to our understanding of pediatric pancreatitis. And on this page are folks that are in Shao in my lab, our WashU collaborators and our external collaborators in Bergen and University of California. Thank you very much for your attention. Oh, okay.
May I have Melina and Ashok also here? We're not taking questions,
Oh,
sorry.
okay.
Talk later.
Okay. Will someone be able to take pictures?
So you're taking them? Or should we be?
All right, maybe we'll take pictures down there
Sure.
too,
Okay.
but. Okay, thank you for the wonderful lecture, Dr. Lowe. So if there are, so we're out of time. So if you have any questions, please ask Dr. Lowe your questions later in the poster session. So this is an award, and it says Frank Brooks Lecture Distinguished Speaker presented to Mark Lowe, MD, PhD, in appreciation of your participation at the APA 2025 annual meeting. So presented.
Go have lunch.